Hey there, science fans! Today we're diving into the amazing world of DNA replication. You know how important DNA is, right? It's like the instruction manual for building you. From your hair color to your height, it's all coded in your DNA. But what happens when your cells need to divide and make more of themselves? How do they make sure each new cell gets a complete copy of that instruction manual? That's where DNA replication comes in. It's like photocopying your instruction manual, ensuring every cell has the instructions it needs. It might sound as simple as pressing a button on a copy machine, but trust me, it's way cooler than that. It involves a cast of amazing characters and some seriously impressive molecular moves. So buckle up, science enthusiasts, as we unravel the secrets of DNA replication. Imagine your DNA molecule like a long, twisted ladder. We call this shape a double helix. Now, for replication to begin, this twisted ladder needs to unwind and separate. Think about trying to read a recipe that's tightly rolled up. You'd need to unroll it first, right? Similarly, DNA needs to unwind so the information in its sequence can be read and copied. But unwinding DNA isn't as easy as it sounds. It's a tightly coiled structure, and separating those strands requires some serious molecular machinery, and that's where our next star player comes in, the enzyme called helicase. Meet helicase, the enzyme that unzips your DNA. Helicase is like a tiny zipper slider, moving along the DNA molecule and breaking the bonds that hold the two strands together. As helicase zips along, it creates a fork in the DNA molecule. Imagine a road forking into two separate paths. This fork in the DNA is aptly called the replication fork, and it's where all the action of DNA replication takes place. The replication fork is a busy place. It's where the two separated strands of DNA are exposed, ready to be copied. Each strand acts as a template, a blueprint for making a new complementary strand. Now, you might think that replication happens in one smooth, continuous motion, right? Well, not quite. DNA replication is a bit more complicated than that, and it has to do with the way the new strands are built. Section 5. Meet the Enzyme Squad, starring Polymerase and friends. Now that our DNA strands are unwound and ready for action, it's time to meet the Enzyme Squad. Leading the pack is DNA Polymerase, the master builder of the DNA world. This enzyme reads the original DNA strand and adds complementary nucleotides, the building blocks of DNA, to create a new strand. But DNA Polymerase is a bit picky. It needs a little helper called primase to get started. Primase lays down a short RNA primer, a starting point, on the DNA template. It's like laying down the first few bricks before building a whole wall. And let's not forget Ligase, the glue that holds everything together. Ligase seals any gaps in the newly synthesized DNA, ensuring a strong and complete double helix. Section 6. Leading and lagging strands, a two-way street. Remember that replication fork? Well, here's where things get a little tricky. DNA polymerase can only add nucleotides in one direction. But since the two strands of DNA run in opposite directions, replication has to happen differently on each strand. On one strand, called the leading strand, replication proceeds smoothly and continuously. DNA polymerase follows helicase along the replication fork, adding nucleotides one after another. But on the other strand, called the lagging strand, replication is a bit more complicated. DNA polymerase has to work in the opposite direction of helicase, creating short, discontinuous fragments of DNA. These fragments are called Okazaki fragments. Section 7. Okazaki fragments, little pieces, big role. Okazaki fragments might be small, but they play a crucial role in DNA replication. Think of them as puzzle pieces that need to be joined together to complete the lagging strand. Each Okazaki fragment is started by primase, which lays down an RNA primer. Then DNA polymerase takes over, adding nucleotides until it bumps into the primer of the previous fragment. Section 8. Joining the dots, Ligase to the rescue. Now we have a bunch of Okazaki fragments on the lagging strand. How do they all get joined together to form a continuous strand of DNA? That's where our friend Ligase comes in. Ligase acts like molecular glue, sealing the gaps between the Okazaki fragments. It removes the RNA primers and replaces them with DNA nucleotides, ensuring a seamless connection between the fragments. Section 9. DNA replication, making copies for life. And there you have it, folks. DNA replication. the incredible process that ensures every cell in your body gets a complete set of genetic instructions. From unwinding the double helix to the teamwork of enzymes like helicase, polymerase, and ligase, it's a symphony of molecular movements. So next time you get a cut or a scrape, remember that deep inside your cells, DNA replication is hard at work, making sure your body can heal and grow. It's a fundamental process of life, and it's pretty darn amazing.